Good afternoon, everyone. I want to thank Tim Hunt and the organizing committee for putting together the conference today. It was an amazing first day. My name is Tom Foti, and I'm the GM of Aldevron's Protein Business Unit. And the title of my talk today is Integrated Gene Editing Solutions for the Cell Therapy Market. So it's been an awesome day because both Tom Whitehead and Victoria Gray are here today, and these people were pioneers over the last 15 years in the cell therapy market. Emily Whitehead and Tom visited our friends in Fargo this past summer, and we all know that she was the first pediatric patient for ALL, which led to Kymriah. Victoria Gray was treated with the first gene-edited cell therapy in Nashville, Tennessee in 2019, which led to Casgevy. And recently, in May of this year, we announced our partnership with Children's Hospital of Pennsylvania. And many of you may have seen that we treated, along with Rebecca and Kieran here that are pictured, baby KJ. It was very exciting because that brought together some technologies in a very expedited manufacturing process. The guide RNA for that project was made by IDT, and they also were a big part of the off-target and on-target data generation for the regulatory agency, and Aldevron made the mRNA base editor that would use as the drug product. So where does this lead to? The Aldevron team, the IDT team are very interested in developing durable, cost-effective, curable cell therapies, and we believe we can do this in an integrated way with our technology stack across our two companies. So if you look at this modality-based way of thinking about workflows, this is how we think about it and plan on addressing and supporting our clients. On the top left, you have ex vivo gene therapy, where we are gene modifying either T cells, HSC cells, NK cells, and clients buy those components from us in a couple of different ways. They either buy them as components, which is the blue line listed up on top, where they buy the guide RNA and the protein, and at the point of care, they make an RNP and gene modify the cells. On the green line, we take one step removed from that process, where we make a GMP RNP, where the client can then just gene modify and take a GMP released RNP, and put it right into their process. And then the bottom modality is this in vivo approach, which was what was done for baby KJ. I also wanted to highlight that we have partnerships with Cytiva, who has a suite of LNPs that can be used for delivery, and I'll talk a bit about that. But in this particular case, then, the guide RNA can be produced by IDT, mRNA drug substance formulated into a drug product and then delivered to our clients. So just to take a little closer look at the tech stack here, if you look on the top right, it's the bolded products. I just wanted to share just a bit about those. And if you look at the legend here, we have in purple off-target editing services, orange is lipids, DNA is in red, blue is RNA and proteins. So if you think about it, a lot of different products have to come together, and that's exactly what we're trying to do, make it easier for our clients to source those from one vendor. On the top is the mRNA process, and it starts with a DNA template. And one of the products that we've come out this year is cell-free DNA. It's called Alchemy. If you're looking to screen a bunch of DNA templates to go into an IVT reaction, you can use that particular technology, and that's highlighted in the red box. In the green box there, we have our IVT enzymes, and those enzymes are vertically integrated into our manufacturing process. The high-cap T7 RNA polymerase is particularly a strong enzyme that it's used both in clinical and commercial programs to date. Everyone is quite familiar with the fact that low double-stranded RNA is very important when you're making a messenger RNA for a therapeutic. And so those enzymes then can be delivered into an IVT reaction, formulated into drug substance with lipids into a drug product. And for base and prime editing on the far right, you can see how that workflow can be very valuable. On the bottom here, we have a protein-based delivery with electroporation. And so in the cell therapy market, this is a workflow that clients appreciate. Again, here we're working with IDT to identify a guide RNA. This is through either their guide RNA library service. They have on-target and off-target nomination services that allow you to pick a guide RNA. It can be combined with our SPI5 or Cas9 proteins, made into an RMP, delivered through electroporation. And when you're knocking in CARs, for example, into different types of cell types, it's really important that you have a high-quality GMP source for your DNA template. Nanoplasmid is a good-selling product for us for knocking in CAR-T. It's been shown to be very effective. And I'm going to show you a little data from University of Wisconsin and how they're using it. IDT recently launched an HDR enhancer protein, so we know that gene editing efficiency is very, very important for knocking in knocking in a DNA template. And this was a research-grade product that was just launched recently by IDT. So this meeting is really about trying to think about forming partnerships. We all have certain expertises, but for us to really advance the field, we really believe, even a large company like Danaher and all its operating companies, we need partnerships. And one of the partnerships that we formed 20 months ago is with the IGI. They have a beacon program with Danaher, where we're trying to advance 2 programs. So I'm going to talk to you a little bit about that, but in this picture is Rainer Blair with Jennifer and Fyodor, and this is what we're trying to do. Despite all this knowledge that we have around gene editing and the clinical tractability, we've only have one therapeutic that's been developed, and we need to get better, and we think we can do that through partnerships. We're working with the IGI and inborn errors of immunity, and there's over 100,000 people that we could treat. There's over 505, and so the plan is to work not only with translational partners like the IGI, but also with regulatory authorities. And so, again, we can't do this alone. One of the things that we learned with this CHOP story and baby KJ was that we need to be in partnership with the regulatory agencies. And there is a vision where they have a platform designation. And this is a great idea for gene editing, because when we're talking about trying to speed things to market and get to clinical trials faster, if you just have to change the guide RNA in the template, it's very helpful from a regulatory standpoint not to go through a full release with the regulatory department, but to maybe have an expedited version. So we're working with the FDA on that, and with their 2 platform designations. So what specifically are we doing with the Beacon? So the idea is to advance 2 programs to phase 1 clinical trial. The one program is focused on SCID, where we're doing a gene modification of HSC cells. The second program is focused on HLH, where we're gene modifying T cells. If you look on the bottom of this slide, you can see all the different Danaher operating companies that are a part of the Beacon. And in the center, you can see all of the talented people at the IGI and at UCSF that are helping with the program. Tomorrow, Fyodor is giving a talk with Sadek at 11 o'clock, and you can hear more about the program there. So another case study in the last few minutes to highlight here is CRISPR genome edited CAR T cell development at the University of Wisconsin. This is a non-viral approach to knocking in a CAR into the tract locus into T cells. So Chris Saha, who is a researcher there, and Dr. Christian Capitini, who is the acting director of the Carbone Cancer Institute, are partnering with Aldevron on this project. And so this is a way for a single-cut, single-step knock-in approach for double-stranded DNA CAR repair. Specifically, they've done something very interesting, and this is the reason these partnerships are very important. What they've done is they've worked with our nanoplasmid engineering team, and they engineered a PAM site into the nanoplasmid so that the RNP can not only be used to cut the genomic DNA and integrate, it also actually linearizes the nanoplasmid and through the same kind of reaction process. And this has allowed for higher knock-in efficiency. So here you can see 1.7-fold increase in positive CAR knock-in from a gene editing standpoint. And the control in this case is a linearized nanoplasmid with a restriction enzyme. So you can learn more about this program with the paper that's referenced here. This just came out in 2025 in Molecular Therapies. So in conclusion, this is the vision. We want to stack the technology from IDT and Aldevron across these workflows in a more seamless way to have a one-vendor approach in a number of different important functional areas. Here I have a few of them listed. Eventually the goal is to have one quality system. When you're working with clients, it's complicated to have multiple MSAs and SOWs. We're working with clients on one set of agreements. Now, the process for manufacturing these components can take a series of months, and so having a central PMO office and access to SMEs is very important. The last thing I'll mention is from a regulatory standpoint, is that supporting our clients through the different phases of regulatory are very important, and we've learned a lot through that entire process. So one, I talked earlier about IDT's ability to help with validation reports for on-target and off-target effects. Those are very important. We also have support in different jurisdictions around the world. We're supporting RNPs that are being administered in Europe, for example. And so, anyways, we recognize that it's an important part of the full value package. So with that, I would like to thank you for your time and attention, and I want to make sure we stay on time so we get you to that social hour. So thank you very much.